Hello, and thank you for attending this virtual meeting. Today I will be talking about highly connected non-redundant microRNAs in breast cancer molecular subtypes. As you know, transcription in breast cancer is one of the many traits of cancer which shows heterogeneity. Actually, transcriptional patterns are quite diverse, and this is the basis of the well-known molecular subtypes, which neatly associate specific gene expression patterns to actual clinical manifestations. One consequence of these differences in transcriptional patterns is that when one uses this type of data to reconstruct co-expression networks, we observed basically topologically diverse networks, which in effect are capturing the differences in the gene regulatory program that is behind each of these cancer manifestations. One of the elements that is associated to these differences in gene expression patterns are microRNAs, also known as MIRs. These are non-coding RNAs, which are, as I previously said, are quite well documented to be involved in the gene expression regulation. So they have been a current focus of cancer research. They seem to play important roles in the development of the different manifestations at the molecular up to the clinical uh, level of cancer. And in particular, they are a current interest of the pharmaceutical industry since they could be potential pharmacological targets. So in past work, we've defined the concept of the Commodore Mirror. So this is a microRNA which in a gene microRNA bipartite co-expression network exhibits a topology in which it is highly connected, which can be measured by the degree, and exhibits non-redundance. So its gene neighborhood is quite unique. Since these Commodore microRNAs are able to control the expression of many different genes, they are also able to, through the control of the expression of set genes, they are able to control the associative functions or sets of biological functions all by themselves. So they seem to be quite interesting in terms of controllability, functional controllability in breast cancer. So our goal in this work was to actually reconstruct these microRNA gene co-expression bipartite networks in order to identify Commodore mirrors that are associated to specific manifestations of breast cancer as seen as molecular subtypes. And our analysis pipeline is quite straightforward. We start with expression data for both for genes and microRNAs obtained from the Cancer Genome Atlas, and then we reconstruct these bipartite networks using an adaptation of the uh, relevance networks method. So basically, we calculate mutual information as a measure of statistical dependencies between expression patterns, and then we select the most significant edges which are above a given threshold. Once we obtain these networks, what we do is we do a topological analysis and we are able to identify the Commodore microRNAs, which are those that exhibit high connectivity, which we measure using the degree above 100, and they are non-redundant. That is, we measure with the redundancy coefficient, and we select those that have redundancy below 0.5. Once we identify these Commodore microRNAs, we do overrepresentation analysis on their gene neighborhood in order to identify those biological processes as annotated in the gene ontology database that are associated to each specific microRNA. And then we go out to the literature and try to validate whether these functions have been previously reported in the context of these microRNAs. So our networks look a little bit like this. We have a microRNA layer, a gene layer, and links represent co-expression, which given what we know about microRNAs, we consider these as putatively regulatory interactions. These are visualization of the networks for the luminal A, luminal B, basal, and HER2 enriched molecular subtypes. It is important for me to highlight that these networks are comparable across a wide range of topological parameters due to the construction of these networks. Uh, we focus mostly on the aforementioned patterns, degree, and redundancy. And what I should highlight here is this upper left quadrant, where we have a very small set of microRNAs, which, as we can tell from the color, are associated to each of the molecular subtypes. These are visualizations of the neighborhoods for these Commodore mirrors for in the luminal A, luminal B, and the basal subtype. Interestingly, the HER2 enriched subtype does not exhibit this type of Commodore mirrors. There are no Commodore mirrors selected for the HER2 enriched subtype, as we can tell from these tables. And in this table, you can also see that the same microRNA is behaving basically different in each of the molecular subtypes. So we analyzed their gene neighborhoods, and we identified a large set of biological processes that are being controlled by these Commodore microRNAs. So we were interested in figuring out what type of functions is each microRNA controlling in each molecular subtype. And what we do is we basically go out to the literature and we automatically search for dimensions of the microRNA alone and dimensions of the microRNA and the representative term or the function. And what we find here is that the set of associated biological functions that we can validate through the literature are functions such as angiogenesis, cell adhesion, apoptosis, gene expression, and signaling, which are basically the hallmarks of cancer. So these functions are associated to those features that make cancer cancer. So to wrap up, we were able to identify these Commodore mirrors for three subtypes of breast cancer, the luminal subtypes A and B, and the basal. Each microRNA is associated to a specific set of genes, and through the putative control of the expression of these genes, they are able to control specific functions, and in particularly functions that are associated to cancer as a whole. Here, I would like to thank my colleagues at INMEGEN, also our funding agencies. And I should point you to the references for this work, as well as the currently available preprint where we discuss in detail the results of our work. And with that, I will open the floor for questions, and thank you very much.